Hello my dear students and my dear friends. I hope you're all doing great, and I'm sure that you have enjoyed the last video that is on RH incompatibility, and if you have any question based on that, please do not hesitate to write down the questions below in the comment box in that particular video. So we'll start here with something very much interesting, and the topic suggests is the blood grouping. Before we get into the blood grouping, few points that a blood is one of the essential features that we have in our body, which is also known as a liquid connective tissue, and a blood is basically made up of the plasma as well as the blood cells. And we have three types of blood cells. We have red blood cells, we have the white blood cells, we have platelets. But here in this video we are not going to talk about the features of the blood cells, neither the features of the plasma. Rather, I have taken something different which is equally interesting and important for your exams, that is a blood grouping. Before we begin with the blood grouping as a whole, let's try to understand few points. What are the things that we are going to cover? And let's take something on an introductory level. Basically, you know what? Till today we have more than 20 types of blood groups that we have in our body. Like we have Duffy's blood group, we have Bombay blood group, there are several examples. But not all the blood groups are clinically significant. Out of all of them, the most significant one, of course the clinically significant one till today, is your ABO blood group as well as the RH blood group. So definitely in this video lecture my focus will be more on ABO and RH. But let me assure you, I will also make a separate video on Bombay blood group. That is something very interesting, and I'll tell you also how people get confused between the Bombay blood group and O blood group. We'll see something, but not here. Before we get into the blood grouping and how the thing is done and everything, we need to understand few terms which are directly associated with the blood grouping. And these are, first is this. We have donor and we have recipient. Suppose if I am a donor, that means I will be donating my blood to the recipient. That means individual number 1, individual number 2. So the donor will donate his or her blood to the recipient. In that case, what we can confirm? The blood of the donor will be transfused to the recipient. So from this individual to that individual, and that transfusion is known as a blood transfusion. I hope that makes sense. Now let's try to understand about the antigen and the antibody. Guys, you see, suppose you are stepping inside the school, and there is one visitor in your school who does not have any idea about who you are, which class you are in, what is your name and all. Can you give me an idea, or can you identify that particular process by which they will come to know everything about you just by looking at something? Simply not by asking, by looking at something. Yes, your guess is right, that will be your eye card. And where do you keep your eye card? Always on your body surface. So my moral of the story is, simply by looking at the eye card, your identity can be identified. Similar way, if you talk about the blood, if you talk about the blood cells, a typical blood cell, they have some identical feature on their surface. Like you are having ID card on your body surface, so they also have their ID card on their body surface, and that ID card is known as the antigen. So antigen is the ID card of the blood. We'll explore more on that when we'll start with the core of the blood grouping system. And if you remember, I told you that along with the blood cells, the blood also contains plasma. So in the plasma of the blood, they have some other proteins as well, which is known as antibody. So definitely we will talk about all of them in detail, and most of you have already known that if there is a reaction between antigen and antibody, that will lead to the agglutination. And since we are talking about blood grouping, that agglutination, if it happens at all, that will lead to the clumping of the blood. So that also we will see when it takes place. Now if we focus here, we have ABO system and the RH system, two most important clinically significant blood group till date. The RH factor or the rhesus factor was first studied in the rhesus monkey, and the discovery of the RH factor was done by Mr. Karl Landsteiner along with Mr. Wiener. These names are important. ABO blood group, that is blood group A, blood group B, blood group O. This system was invented by again Mr. Landsteiner. But if you remember, you have one more blood group, that is the AB blood group. So discovery of the AB blood group was done by Mr. DiCastrillo and Sturley or Sturley, however you pronounce it. I hope this introduction part makes sense to you. Now we will shift directly towards the ABO system and the RH system, and rest of the thing I will wipe out. So we'll start with the ABO system. You know what? Precisely, if we talk about this, if we take ABO system into consideration, we know that we have totally 4 different types of blood groups. Right? So, say we have blood group A, we have blood group B, we have then AB, and finally we have the O, right? But the first question occurs here, on the basis of what we are designing this blood group. Now if you remember, I told you something about the I card. So these blood cells, precisely the red blood cells or the erythrocytes, they have the identity card for the whole blood group as a community. So let me tell you that if I have A as a blood group, that means my red blood cells will be definitely having some particular ID card. If you have B blood group, that means your red blood cells must be having a particular ID card. So what we do with that I card? With the help of all the I card, the blood group can be identified, and that is the function of the identity card, right? So same thing here. The name of that particular ID card here, taking blood group into consideration, is known as your antigen. And as you all know, antigen is a protein. So my moral of the story as of now, if I say I'm having A blood group, that means I will be having a typical antigen as my ID card. And who will be holding the ID card? The red blood cells. Now suppose, now we are actually getting more inside the story. Now if I tell you something about the A blood group, suppose I'm having A blood group. So how the blood group of mine will be identified? Say, like I've told you, this I card will be carried by the red blood cells, neither by the white blood cells nor by the platelets. So taking that into consideration, needless to say that this I card will be present only on the red blood cells. So my first task is to make the red blood cells everywhere, at least in these 4 boxes. So let's make one red blood cells. So red blood cells, red blood cells, red blood cells, and the red blood cells. I've taken one as a whole, right? Now if I have A as a blood group, that time my blood, precisely the red blood cells of my blood, will have antigen known as A. So let's make that first, and let's make in this shape. This. Don't go by the number, it can be n number. Don't go by the number. For the sake of our own understanding, I am making 2 for each. And needless to say that each one of them will be having specific shape and size. So if my blood group is A, I will have antigen A, first thing. I hope that makes sense to you. So definitely, if this is you and you are going inside the school and I am the visitor, and by looking at your own ID card, I can come to know you belong to which class. As simple as that, right? Similarly, you focus here for the B. So if your blood group is B, that means the red blood cells of your blood will carry B antigen. So let's make it here. So this is your B antigen. Can you see that I have made 2 different shapes here? Because they are highly specific in that manner. AB, needless to say, it will carry both the antigen. So let's make it 1. So this is your antigen A, and this is your antigen B. O blood group. He will neither have A antigen, he will nor have B antigen. I hope that makes sense to you. So once again, if you have A as a blood group, on the surface of your red blood cells will have A antigen. If you have B as a blood group, on the surface of your red blood cells will have B as an antigen. If you have AB as a blood group, on the surface of red blood cells you will have both the antigen, and if you have O as a blood group, you will have none of them. I hope that's clear. Right? Now suppose, just imagine, say this is a blood, this portion is the blood. So if this is one of my blood cells, so definitely I will also have plasma here, because if you remember, the major 2 components of the blood is 2, the blood cells and the blood plasma. So I've made the blood cells. I'm not talking about the white blood cells and platelets. Let's assume they are there. Let's assume it. Then why I'm not making them? Because these antigens are only present on the surface of the red blood cells. So red blood cells, as simple as that. So you've got the red blood cells, or precisely you have got the blood cells. Now suppose you have got the plasma. So these are the portion where the plasma will be visible, right? So antigens of blood will be located on the surface of the RBC. And if you remember, we have one more protein which is known as antibody. And these antibody, precisely they are known as immunoglobulin. So these are the globulin protein. You remember we have 4 types of protein in blood: albumin, globulin, prothrombin and fibrinogen. These are one of the 4 major protein factor of your blood. So one of them is a globulin. So here you will find the immunoglobulin, and typically you will have which class of immunoglobulin? You will have immunoglobulin M. So whatever antibody you will have in your blood, it will be of IgM class type. Now let's focus here. What we will get, and let me tell you, this IgM, how do they look like? They look like this. I hope this is visible to you, this part. Yeah. Now let's see how it looks like. If this is an antibody, it will have 5 unit, 1, 2, 3, 4, 5. And not only that, these units are again divided into this fork-like structure. Like this. And this end of this fork, they have complementary shape of the antigen, so that they can have a binding with an antigen. So let's take an example. Suppose this particular antibody is supposed to bind with antigen A. Now see the shape of the antigen A, it looks like triangular. So definitely it will get fit here if I make the shape here as triangular. That makes sense? Point number 1. Think about here, see the shape here, circular. So definitely I have to make it here circular. Then only they will come and they will fit with each other. That is what you will call as a antigen antibody reaction, agglutination. So then only the agglutination will take place, right? So if this antibody is to be bind with the antigen A, that time they will be having a shape of like this. If this antibody is to be bind with the antigen B, they will have a shape like this, as per the diagram. So any one of them they will be having, right? So that is how the antibody looked like. That is how the immunoglobulin M looks like. I hope that makes sense. Now here, if you have A as a blood group, that means you will have A as an antigen and you will have an antibody of B antigen. So the plasma of the A blood group will have anti-B antibody. Antibody, immunoglobulin. Anti-B against the B. Now what is B? B is an antigen. I hope that makes sense to you. That is why if you have A blood group, you cannot donate the blood group to the B. Similar way, if you have B blood group, you cannot donate your blood to the A. Why? Just see here. Suppose you are having B blood group. That means on the surface of your red blood cells you will have B antigen, right? So the moment you are transfusing your blood to me, let's take an example, to me. So what will happen? I have an antibody against your antigen. So what will happen? They will get joined with each other. That will lead to the clumping of the blood. That makes sense, I guess. Similarly, if you have B as a blood group, that means you will be having B as an antigen on the surface of your red blood cells. Along with that, your plasma will have antibody against the antigen A. So how that time it will be known as anti-A antibody. Are you getting my point? See this difference. If you have A as a blood group, you will have A as an antigen. If you have B as a blood group, you will have B as an antigen. First part. Second, if you have A as a blood group, you will have B as an antibody. If you have B as a blood group, you will have A as an antibody. I hope that makes sense. Now you see here, AB. Now this is something very interesting. AB means both the antigen is present on your red blood cell surface. So what do you think? In this scenario, are you able to keep any antibody? No. Why? Suppose if you keep anti-B antibody, what will happen? It will agglutinate here. If you keep anti-A antibody, it will agglutinate here. Right? That is why in AB blood group, you will not find any antibody. Fine? So here, no antibody. Right? O. Yes, you have got it right. In O blood group, both the antibody will be present. That is anti-A antibody anti-B antibody. Again I am saying anti-A means against the A. Now what is A? A is your antigen. So this is your A antigen. So I am preparing an antibody which is against my A. So that will be known as anti-A. So that is my anti-A antibody. Same thing goes for B. I hope that makes sense to you. That's about the ABO system here as of now, keeping antigen and antibody in the mind. So moral of the story, A blood group will have antigen A and antibody against B antigen. So that is anti-B antibody. Right? B blood group will have B antigen and A antibody. You see here, anti-A antibody. AB blood group, since they are having both the antigens, so you cannot keep any of the antibodies. So no antibody here. O blood group, no antigens here. So both the antibodies are present, anti-A, anti-B. I hope that the whole picture is clear to you now. And the antibody, which is known as immunoglobulin, they are of IgM class. I hope that makes sense to you, right? And let me tell you, based on this, the blood transfusion matters, and based on this, the blood mismatch occurs. Now if I see the list, or if I prepare the list of donor and recipient, how it will take place, let's see that. Say so let's write it here, donor, and say this is your recipient. Right? Let's write the blood group here. A, B, AB, O, A, B, AB, O. Right? Now let's see. We'll use two colors for yes and no. So I'll use red for no, and I will use green for yes. Let's see now. A. If A is donating blood to the A. Now see, the blood group of the A is having an antibody not against the A antigen but against the B antigen. If that is the case, the transfusion can be safe, right? I hope that makes sense to you. Once again I am saying, the A blood group has the antibody against the B antigen, not the A antigen. So there will be no question of the transfusion. There will be no question of the agglutination. So there will be no question of the agglutination, right? So easy transfusion can take place. Fine. B blood group. If B blood group donates blood to A, now you see A blood group plasma, if you remember, it contains an antibody against the antigen of B, anti-B antibody. So if you donate it here, what will happen? The anti-B antibody and antigen B will clump together and agglutination will take place. So the transfusion is a big no. It's not possible. AB. If AB blood is being donated to the A blood group, now you think AB is having both the antigen A, antigen B. You come here, A blood group, it is having anti-B antibody, if you remember. So transfusion is a big no. Once again, it's not possible, right? O. If O blood group is being donated to the A, since O does not have any antigen on its surface, no matter what antibody you have, how it will be possible to create a clump? Yes or no? So in that case, O can donate blood to A. So it will be yes. Right? I hope that makes sense now. Fine. B. I hope the first line is clear to you. Now the second part is B. Now you see here, if A blood group is donated to the B, now the plasma of the B is having antibody against A antigen. So there will be chance of agglutination. So transfusion is not possible here. Right? But in case of here, B to B, very much possible. Same reason for this, A to A. So we are not explaining the same again. AB, if AB blood group is being donated to the B, AB contains both the antigen A and B. B is having antibody against the B antigen. So clamping will take place, agglutination will take place. So here also transfusion is not possible. Right? O. Same reason, O can donate blood. Why again? It does not have any antigen. So no matter what antibody you have, clumping will never be possible. Because you have antibody, but you do not have any antigen. Right? Now if we take AB into consideration, A blood group will be giving blood to AB, right? Now AB, they do not have any antibodies. If you remember, they have both the antigens, but they don't have any antibodies. So if they do not have any antibodies, no matter what antigen comes in contact with, it will not create any agglutination. Because for agglutination you need to have two things, antigen and antibody, right? You have antigen here, antigen A on the surface of the red blood cells, but where is the antibody? No antibody. So the transfusion is safe here, right? Same for B. B antigen but no antibody against the B antigen, so again it is safe. AB, same. You have both the antigen here, but where is the antibody? No antibody in the plasma of the recipient, so again no agglutination, so safe transfusion, right? O blood group, no antigen on the surface, no antibody on the plasma. Again I am saying, no antigen on the surface of the red blood cells, no antibody on the plasma of this blood, so it is safe again. I hope that makes sense. O blood group. Now here what happens? If it is O, that means it is having both the antibodies, anti-A antibody, anti-B antibody. Now if you are giving A blood to the O blood group person, so against the A antigen you are having one antibody, anti-A antibody, right? So transfusion is not possible here, right? You come here now. If it is B, again the same thing, against the B antigen you are having anti-B antibody, so again it is not possible, right? AB, both antigens, so both antibodies, a big no, no transfusion, it's not possible at all, it's impossible. O, again no antigens on the surface, both the antibodies on the plasma, but like I've told you, in order to agglutination happen you need to have two things, antigen and antibody. You have got antibodies in the plasma, but where is the antigen on the surface? So here transfusion can be done. So this is the overall chart based on the donor and recipient. But how we did that? Keeping the presence of antigen and antibody in our mind. And we have seen that presence of antigen will be always there on the surface of the red blood cells. Presence of antibody will be always there in the plasma. Right? Now if you carefully observe this chart, one thing you will notice here, that is every blood group like AB and O, can you see that they can donate to their own blood group? Means A can donate to A, B can donate to B, AB can donate to AB, and O can donate to O. In addition to that, you will also observe that O blood group is donating to each one of them. That is why O is known as the universal donor. We all know the fact. AB blood group is accepting blood from all the category AB and O. That is why it is known as universal acceptor, right? So that's about the compatible transfusion of the blood, keeping ABO system in our mind. So as of now we have seen that on the surface of the RBC you can have a protein which is known as antigen, the identity card, which can be A antigen, which can be B antigen, and based on that they are known as A blood group, B blood group, AB blood group, and if none of them are present, O blood group. I hope this is absolutely clear to you now. In the next part we'll start now with the RH system, that is the rhesus factor. Again, that is something interesting now. So let me write it here first. RH factor. Now let me tell you one thing. Let's make an RBC first. If this is an RBC and say the individual's blood group is A, so needless to say I need to have an antigen here, A antigen, right? But whenever we say, or whenever if I ask you rather what is your blood group, and suppose if it is A, you will not say as sir my blood group is A, you will say as my blood group is like A positive or negative, right? Suppose my blood group is AB positive. So every time if somebody asks me, I always say it is AB positive. I don't say it's AB, it's AB positive. Now my dear students, this positive and negative comes into this section, that is the RH factor, which is also known as rhesus factor. So this positivity and negativity can also be identified by the presence of one more protein which is known as rhesus protein, or which is alternatively known as the D antigen. So D is basically for the identification of rhesus factor or RH factor. So you have got A antigen, you have got B antigen for A, B and O if it is not there, and now you have got D antigen for the rhesus factor. Now see, if your blood group is AB positive, that means what? Along with the A antigen you will have one more antigen which will be known as your D antigen. I hope that makes sense to you, right? Now let's take one more example. Suppose this is now AB positive. So you have given A antigen, right? Let's give one more antigen, that is B. So this is your B antigen. So now if you do not see this particular part, this one, now it is my AB blood group, A antigen, B antigen. Now it is given as positive, means I need to have one more antigen on the surface of the RBC. And what is that antigen? It is the D antigen. So if this is a picture given to you, that means I have both the antigens for the ABO system and I have the antigen for the RH system. So my blood group is AB positive, right? Now let's take one more example here. Suppose if I tell you that your blood group is O positive, right? So first you make it O, so RBC. So O means you know that on the surface of the red blood cells of O blood group individual, you will not found any of the antigen. So no A antigen, no B antigen. But can you see this here? It is rhesus positive. So definitely you will have D antigen on the surface. So this is your D antigen. Did you get that? Now if it is O negative, if you write O negative, that means what? You will not have the D antigen on the surface. Did you get that? So that's about the rhesus factor or Rh factor, which is actually denoted by the antigen D. So it is a D antigen. So as of now we talked about three types of antigen: antigen A, antigen B and antigen D. We have seen that for antigen A and B, the plasma of the blood contains its complementary antibody. So suppose if it is A antigen, so it will be anti-A antibody like this will be present. But for Rh factor, for D antigen, blood does not contain any antibody from the beginning. So when it produces when it senses the presence of a D antigen, and that is where the hemolytic disease of the newborn takes place. That is where the Rh incompatibility takes place. If you want to know more about Rh incompatibility, I am giving the video description below in the link. Again, just like the video that I have prepared just before this, go through the video, and I hope the picture about the Rh incompatibility will be clear to you. So anyways, as of now, this picture about the Rh factor, the Rh system, the ABO system is clear to you. And let me tell you one more small fact: worldwide 85% population is Rh positive and remaining 15% population is Rh negative. So that means if I say you are Rh positive blood group or individual, that means you will be having D antigen on the surface of your red blood cells. If I am AB negative, that means I will have A antigen, I will have B antigen, but I will not have D antigen. I hope that makes sense to you now. And let me tell you, in the upcoming series of the video lectures, I will be telling that this ABO system and Rh system, both of these two are genetically determined. That is again something very interesting. So in the next set of video, I will be coming up with the genetical determination of the ABO system as well as the Rh system. Till then, I hope you have enjoyed the video, you have understood the concept of this. So if you have enjoyed the video, press the like button, and if you have not yet subscribed to my channel, subscribe to my channel without fail, so that you can get this sort of videos in near future again. So till then, bye bye.